Right now, as you're hearing this, approximately 15% of the global population is walking around with something in their veins that really shouldn't be there. Something that contradicts evolutionary logic. Something that researchers still can't completely explain. It's called Rh-negative blood, and it stands as one of the most unusual puzzles in human biology. Here's what makes it so peculiar. Every single primate on the planet has the rhesus protein on their red blood cells. Chimps have it. Gorillas have it. Orangutans have it. Every monkey species we've ever examined has it. It's universal throughout our entire evolutionary family tree, except for roughly 15% of humans who simply lack it entirely. They're Rh-negative, and no one knows why. Now, you might be thinking, okay, so some people are missing a protein. Big deal. Genetic variations occur all the time. But this isn't like having blue eyes instead of brown. This isn't some trivial cosmetic difference. This is something foundational. Something that impacts pregnancy, disease resistance, even whether you can receive specific blood transfusions without dying. And the distribution is unusual. In some populations, Rh-negative blood is exceptionally rare. In others, especially among Basque people in northern Spain and southern France, it reaches frequencies approaching 30%. That's not random. That's not natural drift. That's the fingerprint of something else. Something that occurred in our past that we don't comprehend. The Rh factor was only identified in 1940. But the moment scientists understood what they were observing, they recognized they'd encountered something significant, because evolution doesn't typically operate this way. Traits don't simply vanish from entire populations of primates and then appear missing in humans unless there's a reason, a compelling reason. And for 80 years, scientists have been attempting to figure out what that reason is. If you're using this to fall asleep, that's precisely why I create these videos. So, perhaps subscribe and let me know where you're tuning in from and what time it is there. I read the comments and it's fascinating. Tokyo at dawn, London at midnight, Los Angeles at bedtime, all encountering the same mystery together. The theories about this read like science fiction. Some researchers have proposed it's evidence of ancient genetic manipulation. Others point to isolated populations that survived catastrophic events. There are theories about Neanderthal DNA, about ancient bloodlines that trace back to pre-flood civilizations, about genetic advantages that protected certain populations during prehistoric plagues. And then there are the stranger theories, the ones that seem absurd until you start examining the evidence. Theories about extraterrestrial intervention, about the Nephilim referenced in ancient texts, about bloodlines that were somehow different, somehow special, somehow not quite entirely human in origin. Before you dismiss these as conspiracy theories, consider this. Rh-negative mothers can experience immune reactions to their own Rh-positive babies. The mother's body literally treats the fetus as a foreign invader and attempts to destroy it. This is called hemolytic disease of the newborn, and before modern medicine, it claimed countless infant lives. Think about that for a moment. Evolution is supposed to optimize reproduction. So, why would nature create a blood type that makes pregnancy dangerous? Why would a genetic trait persist that actively works against successful childbirth? It doesn't add up unless Rh-negative blood originated elsewhere or evolved under circumstances so exceptional that normal evolutionary rules didn't apply. Throughout history, certain bloodlines have been obsessed with preserving their genetic purity. Royal families intermarried for centuries, creating isolated genetic pools. They called it blue blood, noble blood, blood that was somehow superior, somehow different from common people. Was this merely aristocratic nonsense? Or did they know something about their genetics that we're only beginning to grasp? European royalty suffered from hemophilia at rates that defy statistical probability. Queen Victoria was a carrier. Her descendants spread the bleeding disease across every royal house in Europe. And interestingly, many of these same families had unusually high rates of Rh-negative blood. Coincidence? Perhaps. But the clustering of rare genetic traits in specific bloodlines suggests something more intricate. Over the next few hours, we're going to explore this mystery extensively. We'll trace Rh-negative blood back through history, following the clues through ancient civilizations, medieval dynasties, and modern genetic research. We'll examine the pregnancy paradox that nearly decimated our species. We'll separate the wild theories from the documented facts. We'll look at the claimed superpowers, the medical advantages, the strange correlations that keep appearing in the data. Something occurred in our past, something that left 15% of humanity with blood that shouldn't exist. And the story of how we arrived here is stranger than you can imagine. Vienna, 1937. Karl Landsteiner was already renowned. He'd won the Nobel Prize 6 years earlier for identifying the blood group system, solving one of medicine's greatest mysteries, why some blood transfusions saved lives, while others killed patients within minutes. But now working in his laboratory at the Rockefeller Institute in New York, he was about to stumble onto something that would make his previous discovery seem straightforward. Landsteiner and his colleague Alexander Wiener weren't searching for a new blood type. They were conducting what seemed like routine immunology experiments, injecting blood from rhesus macaque monkeys into rabbits and guinea pigs to study immune responses. Standard procedure, the kind of work that fills laboratory notebooks with predictable results. Except the results weren't predictable. When they tested the antibodies produced by the rabbits against human blood samples, something unusual happened. The antibodies reacted with about 85% of the samples, causing the red blood cells to clump together in that distinctive agglutination pattern that signals an immune reaction. But 15% of the samples showed no reaction at all. Nothing. As if those blood cells were somehow invisible to the antibodies. This shouldn't have been possible. The antibodies were responding to something on the surface of most people's red blood cells, some protein or marker that the majority of humans carried. But a significant minority simply lacked it. They were missing something that most humans possessed. Landsteiner called it the rhesus factor, named after the rhesus monkeys whose blood had helped identify it. If you had the marker, you were Rh positive. If you didn't, you were Rh negative. Simple classification for what would turn out to be an extraordinarily complex mystery. Because here's what made no sense. Blood types exist for a reason. The ABO system, which Landsteiner had already discovered, shows up in other primates. It's ancient, probably evolved to help protect against certain pathogens. But the Rh factor was different. The protein it marked was so fundamental that virtually all mammals carried some version of it. Primates had it. Dogs had it. Even earlier mammalian ancestors had similar proteins. So why didn't 15% of humans? The early research focused on the practical question. What did this mean for medicine? And the answer came quickly, brutally, from obstetrics wards across Europe and America. Doctors had known for decades about a mysterious condition they called erythroblastosis fetalis. Newborns, particularly second or third children, would sometimes be born with severe anemia, their bodies swollen with fluid, their skin tinged yellow with jaundice. Many died within hours. Others suffered permanent brain damage from the buildup of bilirubin in their blood. The pattern was clear but inexplicable. First children were usually fine. Second pregnancies sometimes showed problems. By the third or fourth pregnancy, the risk became terrifying. It was as if something about pregnancy itself was teaching mothers' immune systems to attack their own babies. But what could possibly cause that? The connection to Rh factor came from a pediatrician named Philip Levine in 1939, just 2 years after Landsteiner's discovery. Levine was treating a woman who had suffered a stillbirth. When they tried to give her a blood transfusion to replace what she'd lost during delivery, her body violently rejected blood from her own husband. This shouldn't have been possible. They had the same blood type. Levine tested for the newly discovered Rh factor. The woman was Rh negative. Her husband was Rh positive, and suddenly everything clicked into place. During pregnancy, small amounts of fetal blood sometimes cross into the mother's bloodstream, especially during delivery. If the mother is Rh negative and the baby is Rh positive, inheriting the factor from its father, the mother's immune system encounters a protein it's never seen before. A protein it doesn't recognize as self. So, it does what immune systems are designed to do. It mounts a defense. It creates antibodies. The first pregnancy is usually safe because the immune response takes time to develop. But once those antibodies exist, they don't go away. They circulate in the mother's blood waiting. And if the next pregnancy also produces an Rh-positive baby, those antibodies cross the placenta and begin attacking the fetus's blood cells, treating them as foreign invaders. The mother's body, programmed by millions of years of evolution to protect and nurture her offspring, was being fooled into destroying them. This was unprecedented in human genetics. Yes, other species had immune incompatibilities, but nothing quite like this. Nothing where a mother's second child faced greater danger than her first simply because of blood type. Evolution doesn't usually allow traits that kill babies to persist, especially not traits that specifically target later children when a family is most invested in reproduction. The medical response was swift. By the 1940s, doctors had developed exchange transfusion techniques, replacing an affected newborn's entire blood supply to remove the maternal antibodies. It was brutal, dangerous, but it saved lives. Later came Rh immune globulin, an injection given to Rh-negative mothers after delivery that prevented them from developing antibodies in the first place. Problem solved, medically speaking, but scientifically, the questions were just beginning. Landsteiner died in 1943, just 6 years after his discovery. He never got to see how deep the mystery actually went. Because as researchers began systematically testing populations worldwide, they found something that made no evolutionary sense whatsoever. The Rh-negative blood type wasn't evenly distributed. It clustered. The Basque people of northern Spain showed rates as high as 35% Rh-negative. Other European populations ranged from 15 to 25%, but in Asia, Africa, Australia, the Americas, among indigenous populations that have been isolated for thousands of years, Rh-negative blood was vanishingly rare. In some populations, it didn't exist at all. This geographic distribution suggested something profound. The Rh-negative trait wasn't ancient. It wasn't something humans had carried out of Africa when we first evolved. It appeared to be relatively recent, perhaps emerging in one specific population and then spreading through Europe while remaining rare or absent elsewhere. But when, and more importantly, how? Genetic mutations happen constantly. That's normal, but mutations that eliminate fundamental proteins usually don't persist. They certainly don't reach frequencies of 15 or 20 or 35% of a population. Natural selection should have eliminated any trait that caused maternal immune systems to attack their own babies. Unless the trait provided some other advantage, something so significant that it outweighed the cost of hemolytic disease, something that had allowed Rh-negative individuals to survive, reproduce, and in some populations even flourish. In laboratory meetings and medical conferences throughout the 1940s and 50s, researchers began asking a question that seemed simple but opened doors to extraordinary complexity. Where did Rh-negative blood come from? Some suggested it was simply a loss-of-function mutation, a random deletion of the gene that produces the Rh protein, just one of those things that sometimes happens in evolution. But that didn't explain the geographic clustering or the high frequencies in specific populations. Others wondered if it might have provided protection against some disease, perhaps one that was common in ancient Europe but rare elsewhere. Maybe malaria like the sickle cell trait. But no clear pathogen connection emerged. A few very quietly began suggesting something else. What if Rh-negative blood wasn't a mutation at all? What if it was ancestral, a retention of some earlier human variation that had been lost in most populations but preserved in a few isolated groups? This was speculation, the kind of idea discussed over coffee, not published in journals, but it planted a seed. Because if Rh-negative blood was genuinely ancient, if it represented something from humanity's deep past, then its geographic distribution might be telling us a story about human migrations, bottlenecks, and separate evolutionary paths that we hadn't even begun to understand. The discovery had revealed an anomaly, but the anomaly was pointing towards something much larger, something that would require not just better microscopes and blood tests, but a complete rethinking of human genetic history. The real investigation was just beginning. So, you've discovered that Rh-negative blood is rare. You've learned that it creates medical complications that shouldn't exist if human evolution followed a simple straight line. Now comes the really strange part, the geographic distribution. Because Rh-negative blood isn't evenly scattered across humanity. It's not a random mutation popping up here and there. It follows a pattern, a very specific, very strange pattern that raises more questions than it answers. Let's start with the epicenter. The place where Rh-negative blood reaches its highest concentration anywhere on Earth, a small region nestled in the mountains between Spain and France. Home to a people who have puzzled scientists for over a century, the Basque country. Among the Basque people, 30 to 35% carry Rh-negative blood. Read that again. 1 in 3 Basques, that's not a rare mutation. That's a defining characteristic of the population. To put this in perspective, the global average is about 15%. In most of Europe, it hovers between 15 and 18%. In Africa, Asia, and the Americas, it drops to 1% or less among indigenous populations, sometimes effectively 0. But among the Basques, it's 1 in 3. Now, you might think this is just a statistical fluke, small isolated population, founder effect, genetic drift, all the usual explanations for unusual genetic frequencies, and maybe that's all it is. Except the Basques aren't just unusual for their blood. They're unusual for almost everything about them. Start with their language, Euskara, as they call it. It's like nothing else on Earth. Not Spanish, not French, not related to any Indo-European language, not related to any language family that linguists have ever been able to identify. It stands alone, a linguistic island surrounded by a sea of Indo-European tongues that swept across Europe thousands of years ago, replacing nearly every indigenous language in their path except Euskara. Somehow it survived. Linguists have been trying to classify this language for over 100 years. They've compared it to Caucasian languages, to ancient Iberian tongues, to languages from North Africa. Nothing fits. It's as if the Basques preserved something from a time before the great migrations that shaped modern Europe. Before Indo-European speakers arrived with their bronze weapons and their horses and their conquest, the Basques were already there speaking a language no one else could understand, and they're still there, still speaking Euskara, still carrying Rh-negative blood at rates that defy easy explanation. This isn't just curious, this is a pattern. Genetic isolation paired with linguistic isolation. A people who somehow remain distinct while wave after wave of migration washed across Europe around them. But here's where it gets even stranger. The Basques aren't the only people with elevated Rh-negative frequencies. They're just the most extreme example. When you map Rh-negative blood across Europe, you find a gradient, a clear directional pattern that starts in the Pyrenees and spreads outward. Move west from the Basque country into the rest of Spain and Portugal, and the frequency drops slightly. Still high by global standards, 25 to 27, but lower than among the Basques themselves. Move north into France, and you see similar patterns. Western France, especially Brittany and Normandy, shows frequencies around 25%. These are the Celtic regions, the Atlantic fringe, the parts of Europe that remained somewhat isolated from later migrations. Continue north to Ireland and Scotland, and the pattern holds. The Irish show Rh-negative frequencies around 25%. The Scots, especially in the Highlands and Islands, similar numbers. Wales, roughly the same. All the Celtic regions, all the Atlantic coastal populations, all showing elevated Rh-negative frequencies. Then move east as you travel from the Atlantic coast towards Central and Eastern Europe. The frequency drops, not dramatically, not all at once, but steadily, gradually. Germany shows about 15%, Poland similar. Keep going east into Russia, and it drops further down to 8 or 10% in some regions. Head south into the Mediterranean, and you see another interesting pattern. Greece and southern Italy show moderate frequencies around 15 to 18%, but travel further east into Turkey, and the frequency drops into the Middle East lower still. By the time you reach India and China, indigenous populations show Rh-negative frequencies below 5%. Africa tells an even more dramatic story. In most of sub-Saharan Africa, Rh-negative blood is extraordinarily rare. In some populations, it's effectively absent. Studies of indigenous African populations have found Rh-negative frequencies below 1%, sometimes 0%. It's so rare that when it does appear, researchers have to consider whether it's from recent European admixture, rather than ancient inheritance. The Americas show the same pattern among indigenous populations with no European ancestry. Rh-negative blood is virtually non-existent. Pre-Columbian bloodlines carried almost exclusively Rh-positive genes. The Rh-negative blood you find in the Americas today came with Europeans during colonization mixing into populations that had never known it before. Asia follows suit. Indigenous populations across East Asia, Southeast Asia, and the Pacific Islands show minimal Rh-negative frequencies. Japan less than 1%, China similar, indigenous Australians virtually zero. So, you have this map, this clear, undeniable geographic distribution. High frequencies in Western Europe, especially the Atlantic fringe, a gradient decreasing as you move east and south, near absence in Africa, Asia, and the pre-Columbian Americas. What does this pattern mean? The conventional explanation is migration and mixing Rh-negative, according to mainstream genetics, is just a mutation. It probably arose in one population somewhere, then spread through interbreeding and migration. The high frequencies in Western Europe simply reflect the fact that it arose there or arrived early and became established before later migrations diluted it. The Eastern gradient fits this interpretation. As Indo-European populations moved west from the Pontic steppe around 5,000 years ago, they mixed with existing European populations. In the East, where they started, their genes are more dominant. In the West, they mixed with existing populations that had higher Rh-negative frequencies, creating the gradient we see today. The Atlantic populations, the Basques, Scots, Welsh show the highest frequencies because they were least affected by these eastern migrations. They retained more of the ancient European genetic heritage, relics. Survivors of a genetic pattern that once covered more of Europe before being diluted by incoming populations from the East. This explanation works. It's parsimonious. It doesn't require any exotic theories or mysterious origins, just normal population genetics playing out over thousands of years, but it leaves questions. Why would this particular mutation become so common in Western Europe? Blood-type mutations usually remain rare unless they provide some selective advantage. What advantage did Rh-negative blood provide that could drive it to 30% frequency among the Basques? Why did natural selection favor something that creates potentially lethal complications during pregnancy? And why doesn't the mutation appear in Africa? Humans evolved in Africa. We spent hundreds of thousands of years there before expanding into Europe and Asia. If Rh-negative is just a random mutation, you'd expect to find it everywhere humans exist, probably at similar frequencies, but you don't. It's virtually absent from Africa and only common in one specific corner of the world. Then there's the Basque connection. In North Africa, among the Berber populations of Morocco and Algeria, researchers have found something unexpected: elevated Rh-negative frequencies, not as high as the Basques, but higher than surrounding populations. The Berbers, like the Basques, speak a language that doesn't fit neatly into the major language families of the region. They have cultural traditions that suggest deep roots in North Africa, predating the Arab conquest by millennia. Some researchers have noted physical similarities between Berbers and Basques. Genetic studies show connections, not close connections, not evidence of recent mixing, but deep genetic markers that suggest both populations preserve elements of ancient populations that once inhabited larger territories. You find similar patterns in other isolated mountain communities. The Caucasus Mountains, home to dozens of ethnic groups speaking dozens of languages, show elevated Rh-negative frequencies in some populations. The Atlas Mountains of North Africa, the Scottish Highlands, the Pyrenees. These mountain refuges seem to preserve genetic patterns that vanished from surrounding lands. Mountains protect. They isolate. They create barriers that migrations flow around rather than through. When new populations arrive, they settle the accessible valleys and plains. The mountain people remain, generation after generation, preserving older genetic and cultural patterns. This suggests that Rh-negative might be ancient, really ancient. Perhaps it represents a genetic signature from populations that inhabited Europe and North Africa before the great agricultural migrations, before Indo-European expansion, before the genetic landscape of Eurasia was reshaped by waves of migration over the past 10,000 years. The Basques might be special, not because they developed Rh-negative blood, but because they preserved it. They maintain genetic patterns that existed across a much wider territory in deep prehistory. They're a window into ancient Europe, a genetic museum displaying traits that elsewhere were diluted or lost. But this raises another question. If Rh-negative blood is ancient, if it predates the agricultural migrations, why is it beneficial? What environmental pressures would maintain it at such high frequencies? What were our ancestors facing 15,000 or 20,000 years ago that made Rh-negative blood advantageous enough to reach 30% in some populations? The Ice Age, Europe during the last glacial maximum, was a harsh place. Small populations struggling to survive in a landscape dominated by cold and scarcity. Under those conditions, any genetic variant that provided even small survival advantages could rise to high frequencies quickly. Small population plus strong selection equals rapid genetic change. Maybe Rh-negative provided some advantage we don't fully understand. Maybe it was just genetic drift in small isolated populations, random fluctuations that happened to push this trait to high frequencies. Or maybe there's something else, some piece of the puzzle we haven't found yet. Because the map doesn't just show where Rh-negative blood is, it shows where it survived. And sometimes what survives in the isolated places tells you something important about what once existed everywhere. The Basques remain, the Berbers remain, the mountain populations and Atlantic islanders remain. All carrying in their blood an echo of something ancient, something that refuses to be mapped completely, refuses to fit neatly into our models, refuses to surrender all its secrets to our genetic analysis. The geographic pattern is clear. The explanation is not. And that's what makes this mystery so compelling. Because maps don't just show you where things are. Sometimes they show you where to start digging. Now, here's where the story gets really strange. Because while scientists were mapping Rh-negative blood across populations, discovering its peculiar geographic patterns, someone else noticed something odd. The crowned heads of Europe, those carefully preserved bloodlines that had ruled for centuries, they had a secret and it was written in their blood. The term blue blood didn't come from nowhere. In Spanish, sangre azul originally referred to the pale-skinned Castilian nobility whose veins appeared blue beneath their skin, supposedly proving they hadn't mixed with darker-skinned Moors. But the obsession with blood purity among European royalty went far deeper than skin color. They believed something fundamental ran through their veins, something that made them different from common people, something that justified their right to rule. They might have been more correct than they realized, just not in the way they thought. When researchers began systematically analyzing blood types in royal families during the 20th century, they found something unexpected. Rh-negative blood appeared with unusual frequency among European nobility. Not universal, not even a majority, but statistically significant enough to raise eyebrows. The Romanovs, the Windsors, various branches of Spanish and French royalty all showed higher rates of Rh-negative blood than the general populations they ruled. Now, before we go too far down this path, we need to be clear about something. Correlation is not causation. The prevalence of Rh-negative blood in royal families doesn't mean Rh-negative people are somehow naturally suited to rule. It doesn't mean there's anything inherently royal about this blood type. What it does show is the effect of centuries of deliberate inbreeding of a relatively small population pool, marrying exclusively within itself, concentrating certain genetic traits through sheer mathematical probability. But that doesn't make the story any less fascinating because what happened to these bloodlines, the catastrophes and tragedies that befell them, tells us something important about the dangers of genetic isolation, about what happens when you try too hard to keep blood pure. Let's start with Queen Victoria. Born in 1819, she became the most powerful monarch in the world, ruling an empire that spanned a quarter of the globe. She had 9 children, married them strategically into every major royal house in Europe, and inadvertently spread a genetic time bomb that would help topple dynasties and reshape history. Victoria was a carrier of hemophilia B, a rare genetic disorder that prevents blood from clotting properly. The gene sits on the X chromosome, which means women can carry it without showing symptoms, but their sons have a 50% chance of inheriting the full disease. For someone with hemophilia in the 19th century, even a minor cut or bruise could be fatal. There was no treatment, no cure, just endless vigilance and the constant terror of bleeding to death from injuries that would barely slow down a normal person. Victoria passed the gene to at least 3 of her 9 children. Her son Leopold died from hemophilia at age 30 after a minor fall. Two of her daughters, Alice and Beatrice, became carriers and spread it further. Alice's daughter Alexandra married Tsar Nicholas II of Russia and gave birth to Alexei, the heir to the Russian throne. Alexei's hemophilia changed history, not because of genetics, but because of desperation. Nicholas and Alexandra, watching their only son suffer through constant bleeding episodes, became obsessed with finding someone who could help him. They found Grigory Rasputin, a Siberian mystic who seemed able to ease the boy's suffering, probably through hypnosis and calming techniques that reduce stress-related bleeding episodes. Rasputin's influence over the royal family became scandalous during World War I. While Nicholas was away commanding the army, Alexandra relied heavily on Rasputin's advice, even in political matters. The Russian aristocracy watched in horror as a peasant mystic effectively controlled access to the throne. His presence became a symbol of royal corruption and incompetence. When Rasputin was finally assassinated in 1916, it was too late. The damage was done. The monarchy's credibility was shattered. Less than 2 months later, revolution swept through Russia. Nicholas abdicated, the royal family was imprisoned, and in July 1918, in a basement in Ipatov House, the entire family was executed. Alexei was 13 years old. Would the Russian Revolution have happened without hemophilia? Almost certainly. The social and economic conditions that led to it were far too deep for one family's genetic disease to determine, but hemophilia shaped how it happened, accelerated the timeline, gave revolutionaries a perfect symbol of royal decadence, a child who couldn't inherit his father's empire because his blood couldn't clot, and it all traced back to Queen Victoria's genes spreading through the interconnected web of European royalty like a genetic echo of that ancient bottleneck we talked about earlier. Small population, limited breeding pool, recessive genes finding expression because the same genetic variants kept appearing on both sides of family trees. The Spanish royal family experienced something similar, though even more dramatic. The Spanish Habsburgs had practiced cousin marriage for so long that by the time Charles II inherited the throne in 1665, he was more inbred than a child born to siblings. Genetic analysis of the Habsburg family tree shows he had an inbreeding coefficient of 0.254, which is just extraordinary. To put that in perspective, anything above 0.25 is generally considered incompatible with viability in animal breeding programs. Charles had the Habsburg jaw, a severe prognathism that made eating and speaking difficult. He was reportedly sterile, suffered from epilepsy, and was probably intellectually disabled. He died at 38 without heirs, and his death triggered the War of Spanish Succession that killed hundreds of thousands across Europe. This wasn't hemophilia. This was the accumulated burden of multiple recessive genetic disorders, all expressing simultaneously because the family tree had stopped being a tree and become more like a decorative wreath, loops connecting back to themselves over and over. But, here's what makes the Rh-negative connection interesting. These families weren't just concentrating disease genes. They were concentrating all their genetic traits, including blood type. If one or two early royals happened to be Rh-negative, and their descendants kept marrying each other, the frequency of Rh-negative blood would naturally increase in their lineage, not because it made them royal, but because math works the same way, regardless of how fancy your crown is. Some researchers have speculated that Rh-negative blood might have contributed to the high infant mortality rates in royal families. Remember the pregnancy paradox we discussed earlier? Rh-negative mothers can have serious complications with Rh-positive babies, especially in second and subsequent pregnancies. In an era before modern medicine, these complications were often fatal for both mother and child. Royal families kept detailed genealogical records, but they didn't record blood types. We can sometimes determine them through genetic analysis of preserved remains, but the data is incomplete. What we do know is that royal infant mortality was shockingly high, even compared to their equally privileged but less inbred aristocratic peers. Miscarriages, stillbirths, and children who died in infancy or early childhood appeared with disturbing frequency. How much of this was Rh incompatibility versus general inbreeding depression versus simple historical childhood mortality rates? We can't say for certain. The factors are too intertwined, the data too fragmentary. But the possibility exists that the very blood these families prized as marking their superiority was also contributing to their dynastic instability. There's a certain poetic irony in that. These families spent centuries obsessing over bloodline purity, arranging marriages based on genealogy rather than genetics, which they didn't understand. They thought they were preserving something precious, keeping their royal essence undiluted. Instead, they were conducting multi-generational experiments in the dangers of genetic isolation, proving that evolution requires diversity to function properly. The mystique of blue blood persists even today, long after the divine right of kings has been discredited. You still hear people talk about certain families having something special in their lineage, some inherent quality passed down through generations. And when those families happen to have Rh-negative blood, which some do, it feeds into modern speculation about what Rh-negative blood really means. But the actual lesson from royal bloodlines is almost the opposite of what the mystique suggests. It's not that concentrated bloodlines create superiority. It's that genetic isolation, even among the most privileged humans in history, creates vulnerability. The Habsburg jaw, hemophilia spreading through Victoria's descendants, the constant pregnancy complications and infant deaths. These weren't signs of special blood. They were warning signs written in genetics, evidence that humanity's strength has always come from diversity, not purity. The royal families of Europe were in a strange way conducting their own version of a genetic bottleneck. Not as severe as the one our ancestors survived 930,000 years ago. Not as desperate, but following the same mathematical principles. Small population, restricted breeding, genetic drift concentrating both beneficial and harmful traits in unpredictable ways. And just like that ancient bottleneck, the royal experiment revealed something important about human biology. We're not designed for isolation. Our survival depends on populations mixing and sharing their genetic heritage. The very thing these royal families feared most, the dilution of their bloodline, was actually the thing that could have saved them from their genetic destiny. So, when you hear about blue blood and royal lineages, remember what it really represents. Not superiority, but a cautionary tale about what happens when any group, royal or otherwise, becomes too insular, too convinced of its own specialness to recognize the basic biological truth that diversity is strength. But that still leaves us with questions about Rh-negative blood itself. If it's not a mark of royalty, what is it? Where did it really come from? And why does it persist in modern populations at the frequencies we observe? To answer that, we need to go even further back. So, where did it come from? This blood type that appears in only 15% of the global population, but reaches 40% in some isolated groups. This genetic marker that doesn't follow the normal rules that creates problems during pregnancy, that seems to cluster in specific populations for no obvious evolutionary reason. The mainstream answer is simple: random mutation, genetic drift. Nothing special, just one of thousands of variants that emerge during human evolution and happen to stick around in certain populations. But that answer has never satisfied everyone. Because Rh-negative isn't distributed randomly. It's clustered, concentrated, and the pattern of that concentration has sparked theories ranging from plausible to extraordinary to downright bizarre. Let's start with the most scientifically grounded hypothesis, the Cro-Magnon theory. Around 45,000 years ago, a group of humans entered Europe. We call them Cro-Magnons, though that's really just an archaeological label for the first anatomically modern humans in Europe. These weren't Neanderthals, those had been there for hundreds of thousands of years, perfectly adapted to Ice Age conditions. These were newcomers, migrants from Africa and the Middle East, carrying with them all the innovations that modern humans had developed. Advanced stone tools, sophisticated hunting techniques, art, music, symbolic thinking. When they arrived, Europe was already occupied. The Neanderthals had been living there successfully for over 200,000 years. They were robust, powerful, perfectly suited to the cold climate. They had their own culture, their own technologies, their own social systems. The two species coexisted for thousands of years. We know this because we can see it in the archaeological record. Neanderthal sites and Cro-Magnon sites exist side by side, sometimes within a few miles of each other, and we know they interbred. Genetic analysis has proven this beyond doubt. Modern Europeans carry 1 to 4% Neanderthal DNA. The mixing happened multiple times in multiple places over thousands of years, but here's where it gets interesting. Some researchers have suggested that Rh- might be one of the genetic legacies of that ancient interbreeding. A marker that emerged in those early European populations, either from Neanderthal genes or from the unique selective pressures that Cro-Magnons faced adapting to European environments. The evidence is circumstantial, but intriguing. Rh- reaches its highest frequencies in the Basque region of Spain and France, areas that also show unusual genetic isolation and deep ancestral roots. Some studies have suggested the Basques represent one of the most genetically ancient populations in Europe, possibly direct descendants of those first Cro-Magnon settlers with minimal mixing from later migrations. If Rh- emerged in those early European populations, the Basques might have preserved it at high frequencies simply because they remained relatively isolated for thousands of years. But the Cro-Magnon hypothesis, reasonable as it sounds, doesn't explain everything. Why would a trait that creates pregnancy complications persist at such high frequencies? Natural selection should have eliminated it, or at least reduced it to rare mutation status, unless it provided some advantage, some benefit that outweighed the reproductive costs. Which brings us to theories that venture further from mainstream science. One of the most persistent involves Atlantis. Yes, that Atlantis, the legendary advanced civilization described by Plato, supposedly destroyed in a single catastrophic day and night. Now, most historians consider Atlantis either pure fiction or a philosophical allegory, but that hasn't stopped theorists from connecting it to Rh- blood. The logic goes like this. If Atlantis existed and was destroyed around 11,000 years ago, as Plato claimed, the survivors would have dispersed to other lands. Some theorists point to the Basque region, to Ireland, to parts of North Africa as possible refuge sites, places where Atlantean survivors might have settled, intermarried with local populations, and passed on their genetics, including perhaps an unusual blood type that had been common in their lost homeland. There's no archaeological evidence for any of this. No sunken cities with advanced technology. No genetic markers that can be traced to a single catastrophic diaspora 11,000 years ago. But the theory persists because it offers something the mainstream explanation doesn't. A narrative, a story that makes Rh- blood feel special, connected to something greater than random mutation. Then, there's the ancient astronaut theory. This one pushes the boundaries of speculation even further. The basic premise, at some point in human prehistory, extraterrestrial beings visited Earth and interbred with humans. The offspring of these unions carried genetic markers that distinguish them from purely terrestrial humans. Rh- blood, according to this theory, is one of those markers. Proponents point to ancient texts that describe gods coming down from the sky and mating with human women. They reference the unusual properties of Rh- blood. The pregnancy complications, the claimed differences in physiology and psychology. They ask, why would evolution create a blood type that causes problems unless it wasn't evolution? Unless it was introduced from outside. The evidence cited includes ancient artwork showing figures in what look like spacesuits, megalithic structures built with technology that shouldn't have existed, mythological accounts of sky gods with superhuman abilities, and always the question, if Rh- is just a random mutation, why does it cluster in populations descended from regions with the strongest traditions of divine intervention and heavenly bloodlines? Mainstream science dismisses this entirely. There's no physical evidence of ancient extraterrestrial contact. The pregnancy complications associated with Rh- can be explained through normal evolutionary mechanisms. The clustering patterns match what we'd expect from population genetics and migration patterns. But dismissal hasn't killed the theory. It remains one of the most popular explanations in alternative research circles. More grounded, but still controversial, are the biblical interpretations. The book of Genesis mentions the Nephilim described as the offspring of the sons of God and the daughters of men. The text is brief and cryptic. There were giants in the earth in those days, and also after that when the sons of God came in unto the daughters of men, and they bare children to them. Who were these sons of God? Angels. Fallen divine beings. Members of an earlier, more advanced human civilization. And who were their children? Some researchers have connected the Nephilim to royal bloodlines, to the divine right of kings, to the obsession with pure bloodlines that characterized ancient monarchies. They point to the fact that European royalty, who claim descent from gods and heroes, show higher frequencies of Rh-negative blood than general populations. They note that many ancient cultures practiced strict endogamy, marrying within narrow elite groups, supposedly to preserve sacred bloodlines. What were they trying to preserve? What genetic legacy were they protecting? The biblical account continues with the flood, a catastrophe that destroys the Nephilim and most of humanity. But some survived. Noah and his family, carefully chosen, carrying forward specific bloodlines into the post-flood world. Did those survivors carry Rh-negative blood? Is that why it appears in populations descended from the Middle East and Mediterranean, the region where biblical events supposedly occurred? Again, mainstream scholars see this as reading far too much into ancient texts. The Nephilim account is brief, unclear, and open to multiple interpretations. It might refer to a warrior class, to members of a different tribe, to legendary heroes whose stories got incorporated into religious literature. Connecting it to a specific blood type requires assumptions that the text itself doesn't support. Yet, the mystery remains. Why does Rh-negative cluster in populations with deep mythological traditions of divine ancestry? The Basques, the Irish, the Scottish Highlands, all areas with Rh-negative frequencies above 25%. All areas with rich traditions of fairy folk, divine bloodlines, people who are somehow different from ordinary humans. Coincidence, confirmation bias, or faint genetic memory of something that really was different? The mutation theory offers the simplest explanation. Around 35,000 to 40,000 years ago, somewhere in human populations, probably in Europe or the Middle East, a genetic mutation occurred. The RHD gene, which normally codes for the D antigen on red blood cells, got deleted or inactivated in one individual. That person became the first Rh-negative human. The mutation got passed to their children. If the family lived in a small, relatively isolated population, genetic drift could increase the frequency of the mutation just by chance. Over thousands of years, as populations moved and mixed, the mutation spread to certain groups while remaining rare in others. This is exactly how most genetic variants work. Random mutation creates variation. Genetic drift and population structure determine how that variation spreads. Natural selection then either eliminates it, maintains it, or increases it based on whether it helps or hurts survival and reproduction. In the case of Rh-negative, the pregnancy complications would create mild negative selection pressure, keeping it from becoming too common but not strong enough to eliminate it entirely, especially if it provided subtle benefits in other areas. Modern genetic research supports this explanation. We can trace the RHD deletion back tens of thousands of years. We can see it spreading through European populations during the Neolithic period. We can map its frequency changes over time as populations migrated, mixed, and sometimes remained isolated. It all fits standard population genetics models. No need for ancient astronauts, or lost civilizations, or divine intervention. But even mainstream researchers admit there are puzzles. Why has natural selection allowed Rh-negative to persist at such high frequencies in some populations despite the pregnancy complications? Are there hidden advantages we haven't identified? Some studies have suggested Rh-negative individuals show slightly different immune responses, possible resistance to certain diseases, maybe even subtle neurological differences. But the evidence remains preliminary, contested, difficult to replicate. And there's the psychological dimension. Across decades of research, anecdotal reports keep surfacing of Rh-negative individuals feeling different, experiencing unusual sensitivities, claiming abilities that others don't have. These reports are dismissed by science as confirmation bias, as the human tendency to see patterns that aren't really there. But they persist generation after generation, culture after culture, creating a folklore around Rh-negative blood that science struggles to fully explain or eliminate. So we're left with competing narratives. The mainstream scientific explanation, random mutation, genetic drift, nothing special. The archaeological hypothesis, ancient European populations, possibly with Neanderthal mixture, developing unique genetic profiles. The mythological theories, Atlantis, Nephilim, ancient astronauts, stories that can't be proven but can't quite be silenced. And underneath it all, the lived experience of millions of Rh-negative individuals who insist there's something more to this story than mainstream science acknowledges. The truth is probably simpler than the exotic theory suggests, but more complex than the standard mutation explanation admits. Human genetics is messy. Our history is complicated. And sometimes the most interesting questions aren't the ones we can answer definitively, but the ones that keep us searching, wondering, trying to understand where we came from and what makes us who we are. But carrying this rare blood came with a price that wouldn't become clear until a woman with Rh-negative blood decided to have children. Here's where the story gets dark. Here's where evolution seems to have made a mistake so fundamental that it should have eliminated Rh-negative blood from the human population entirely. Because under the right circumstances, or rather the wrong ones, an Rh-negative mother's immune system could kill her own baby. It works like this. Imagine a woman with Rh-negative blood, O-negative, A negative, doesn't matter which type, as long as she lacks that Rh protein on her red blood cells. She meets a man, falls in love, gets pregnant, and if that man happens to be Rh positive, which statistically he probably is since 85% of people are, then the baby has a good chance of inheriting his Rh-positive blood. Now you've got an Rh-negative mother carrying an Rh-positive baby. For the first pregnancy, things usually go fine. The mother's immune system doesn't know about the baby's different blood type. The placenta does a remarkably good job of keeping maternal and fetal blood separate. It's like two separate circulation systems running side by side, exchanging oxygen and nutrients through cellular barriers, but never actually mixing usually. But during childbirth, those barriers break down. The placenta separates from the uterine wall. Small amounts of the baby's blood enter the mother's circulation. And for the first time, the mother's immune system encounters those foreign Rh proteins on her baby's red blood cells. Her immune system does what immune systems do. It recognizes a foreign invader and mounts a response. It creates antibodies, specialized proteins designed to attack and destroy anything carrying that Rh marker. The first baby is usually born healthy, completely unaffected, but the mother has been sensitized. Her immune system now has a memory of that foreign protein. It's ready. Then she gets pregnant again, another Rh-positive baby, and this time those antibodies are waiting. They're small enough to cross the placenta, small enough to enter the baby's circulation. And once they're in there, they start attacking the baby's red blood cells, binding to those Rh proteins and marking the cells for destruction. The medical term is hemolytic disease of the newborn. The simple translation is that the mother's immune system is destroying her baby's blood while the baby is still in the womb. The baby's body tries to compensate. It produces more and more red blood cells, working desperately to replace the ones being destroyed, but it can't keep up. The baby becomes profoundly anemic. The heart enlarges, trying to pump harder to deliver oxygen with fewer red blood cells. Fluid accumulates in the body cavities. The liver and spleen swell as they work overtime trying to produce replacement blood cells. In severe cases, babies were born swollen, pale, barely alive. Many died within hours or days. Others survived but suffered permanent brain damage from the lack of oxygen during critical developmental periods. Some mothers watched baby after baby die, never understanding why their bodies kept betraying them. Before 1968, doctors could only watch. They understood the mechanism. They could predict which pregnancies were at risk, but they had no way to prevent the sensitization, no way to stop a mother's immune system from attacking her own children. The grief was unfathomable. Imagine being a woman in the 1950s. You have your first baby and everything is perfect. Then you get pregnant again, and this baby dies shortly after birth. The doctors can't really explain why. They use terms like blood incompatibility and immune reaction, but they can't do anything about it. You try again. Another pregnancy, another dead baby, and another. Some families lost 5, 6, 7 children in succession, each pregnancy becoming an exercise in hope against experience, each birth bringing not joy but dread. The historical records from this period are haunting. Medical journals from the early 20th century document case after case, family after family torn apart by something they couldn't see or understand. Parish records show children born and buried within days, sometimes with notes indicating blood disease or simply failure to thrive. And here's what makes it even more puzzling from an evolutionary perspective. This isn't a rare disorder affecting a handful of people. Rh incompatibility is common in populations where Rh-negative blood is frequent. This would have affected thousands of pregnancies every generation. Yet, Rh-negative blood persisted generation after generation despite the obvious reproductive disadvantage. Evolution should have eliminated it. Natural selection is ruthless about removing traits that reduce reproductive success. And what could reduce reproductive success more effectively than having your immune system kill your own offspring? Unless there was an advantage, something that offset the terrible cost. Some researchers have suggested that Rh-negative individuals might have enhanced immune responses to certain pathogens. The theory goes that the same immune system characteristics that make Rh-negative mothers react to Rh-positive babies might also make them more effective at fighting off specific infections. There's some evidence for this. Studies have found that Rh-negative individuals show different susceptibility patterns to certain diseases, though the data is still incomplete. Others have proposed that Rh-negative blood might offer protection against certain parasites. Toxoplasmosis, a parasitic infection that can cause severe problems during pregnancy, seems to affect Rh-positive and Rh-negative individuals differently. Some studies suggest Rh-negative people might have enhanced resistance, though again the evidence is mixed. Or maybe the advantage isn't biological at all. Maybe it's about population structure. Remember, Rh-negative blood is most common in isolated populations, particularly in the Basque region and parts of Western Europe. In small, relatively isolated groups, genetic drift can preserve traits that might otherwise be selected against. If a trait becomes common in a small population just by chance, it can persist for many generations, even if it has some costs, as long as those costs aren't immediately catastrophic. But, for most of human history, those costs were indeed catastrophically high for the affected families. Then came 1968. Two scientists, Vincent Frieda and John Gorman in New York and William in Liverpool, working independently, developed a treatment. They called it Rh immunoglobulin. We know it now as Rogum. The concept was elegantly simple. If you could prevent the mother's immune system from ever recognizing those Rh-positive blood cells during the first pregnancy, you could prevent sensitization. No sensitization, no antibodies. No antibodies, no attacks on subsequent babies. Rogum is essentially a dose of pre-made antibodies against the Rh protein given to an Rh-negative mother shortly after delivery of an Rh-positive baby. These antibodies quickly bind to any fetal blood cells that entered her circulation during childbirth. They coat those cells, hiding them from the mother's immune system. Before her body can mount its own immune response, the foreign cells are cleared away and the pre-made antibodies break down naturally. Her immune system never learns to recognize the Rh protein, never develops the dangerous memory, never creates its own antibodies. The next pregnancy can proceed as safely as the first. The impact was immediate and dramatic. Within a decade, deaths from hemolytic disease of the newborn dropped by more than 90% in countries where Rogum became standard care. Families that would have been torn apart by successive infant deaths could now have as many healthy children as they wanted. Today, the protocol is routine. Every pregnant woman gets her blood type checked. If she's Rh-negative and the father is Rh-positive or unknown, she receives Rogum around 28 weeks of pregnancy and again after delivery if the baby's confirmed Rh-positive. Additional doses after any event that might cause fetal blood to enter maternal circulation, miscarriage, amniocentesis, abdominal trauma. It's become so routine that most people don't even think about it. A simple injection, a few minutes, but that injection represents one of medicine's greatest triumphs over biology. A solution to a problem that had plagued humanity for potentially hundreds of thousands of years because here's the thing. This pregnancy paradox wasn't new. It didn't emerge with modern medicine or modern lifestyles. It's been happening for as long as Rh-negative and Rh-positive humans have been having children together. Every generation, every century, every millennium since the Rh-negative trait first appeared in human populations. Before modern medicine, before we understood blood types, before we even knew blood circulated through the body, mothers were losing babies to this invisible war between immune systems. They blamed bad luck, divine punishment, curses, hereditary weakness. They had no way of knowing that the problem was written in their blood in the presence or absence of a single protein. And yet despite this, despite the grief and loss, despite the clear reproductive disadvantage, Rh-negative blood survived, persisted, remained part of the human genetic legacy, which brings us back to the central mystery. What advantage could possibly offset such a devastating cost? What benefit could be worth the price of watching child after child die? We don't know for certain. Maybe we never will. But the fact that Rh-negative blood is still here, still common in certain populations, still being passed from generation to generation, suggests that somewhere in our evolutionary past, it provided something valuable, something that made it worth keeping despite the terrible price. Or, perhaps the answer is simpler and sadder. Perhaps there was no advantage at all. Perhaps Rh-negative blood is simply a neutral trait that happened to become common in certain populations through chance. And perhaps thousands of families paid the price for that accident of genetics, suffering losses that served no purpose, taught no lesson, provided no benefit. Either way, thanks to Rogum, that suffering has mostly ended. The pregnancy paradox has been solved, at least in places with access to modern medical care. An Rh-negative mother can now carry an Rh-positive baby with no more risk than any other pregnancy. But the trait remains. The blood type persists, and the questions it raises about human evolution, human migration, and human survival continue to fascinate researchers even today. So, now we arrive at the part of the story that keeps this topic alive on late-night forums and whispered family conversations. Not chromosomes, not antibodies, but superpowers. Because once people realized there was a blood factor that most humans had, and a small minority didn't, the imagination did the rest. From there, an ordinary biological quirk slowly turned into a catalog of alleged abilities: lower blood pressure, higher intuition, strange eye colors, sensitivity to sunlight, immunity to certain illnesses, even psychic gifts. One by one, these stories began to attach themselves to Rh-negative blood, like magnetized filings clinging to a small but noticeable anomaly in the human story. Let's walk through them. Not to mock them, not to blindly accept them, but to see where evidence ends and folklore begins. Imagine a quiet room, a questionnaire, a group of people with Rh-negative blood, and another group with Rh-positive. You ask them how they feel in their bodies, how they move through the world. The first cluster of claims is physical. More often than not, you'll hear that Rh-negatives tend to have lower blood pressure, lower resting body temperature, heightened sensitivity to pain, unusual reactions to heat or cold, and a kind of oversensitivity to bright light or direct sunlight. Some people describe walking into the sun and feeling almost overwhelmed, as if their skin and eyes are amplifying the light rather than filtering it. Others say they faint more easily in hot rooms or are the first to reach for a sweater when the temperature drops. In online communities, these shared experiences become patterns. Patterns become narratives. Before long, the story congeals. Rh-negatives are physically different, more delicate in some ways, more tuned in others. There is a kernel of reality in some of this. A few small studies have suggested that, on average, people with Rh-negative blood might have slightly lower blood pressure than Rh-positive. The differences are modest and not all studies agree, but the idea is not entirely invented. Light sensitivity is similar. Many people who self-identify as light-sensitive or sun-sensitive also happen to be fair-skinned with lighter eyes, traits that cluster in some of the regions where Rh-negative is more common. Blue or green eyes, pale skin, Rh-negative blood. They travel together in certain populations. It's easy to connect the dots and assume the blood type is the driving cause when all of them may simply come from the same ancestral gene pool. That brings us to one of the most persistent claims: unusual eye colors. Scroll through Rh-negative forums and you'll see it repeated like a mantra. We have hazel eyes, green eyes, eyes that change color. We don't fit standard categories. There are stories of eyes shifting shade with mood, light, even health. To some, this becomes more than pigmentation. It becomes a sign, subtle visible proof that their blood marks them as different. Genetically, eye color is controlled by multiple genes, many of which are common in the very European and North African populations that also show high Rh-negative frequencies. So, the link between Rh-negative and lighter, mixed eye colors exists statistically in some groups, but it's a side effect of shared ancestry, not of the Rh factor itself. Still, when you look in the mirror and see unusual eyes staring back, and you also know you belong to a rare blood group, it's hard not to weave them together into a single story. Then there is intelligence. Many Rh-negative people report being told they are gifted, that they read early, that they think in unconventional ways. Others point to small studies, often misquoted or taken out of context, claiming that Rh-negative score higher on IQ tests. This is a powerful idea. The thought that your blood type secretly encodes a sharper mind can be flattering, reassuring, even comforting if you've always felt out of step with those around you. But when researchers look carefully at large populations, controlling for education, socioeconomic status, and region, the alleged IQ gap largely dissolves. Any differences are small, inconsistent, and rarely rise above statistical noise. Intelligence is complex. It's shaped by genes, yes, but also nutrition, childhood environment, schooling, health, even luck. There is no robust scientific evidence that the simple presence or absence of one antigen on red blood cells makes you meaningfully smarter. And yet, the myth persists not because it's been rigorously demonstrated, but because it speaks to a feeling many Rh-negative people already have. That they think differently, feel differently, and are searching for an explanation. Health claims go deeper still. Some believers insist that Rh-negative blood provides resistance against certain viruses or parasites. You'll see claims that Rh-negatives rarely get HIV, or that they are less vulnerable to malaria, toxoplasmosis, even to some respiratory infections. The reality is nuanced. There is evidence that different blood groups, including the ABO system and the Rh factor, can influence susceptibility to certain diseases. For example, some ABO types have been linked to specific cancer or cardiovascular risks. Rh status has been studied in relation to Toxoplasma gondii, the parasite carried by cats. A few studies suggested that Rh-negative and Rh-positive individuals may show different patterns of reaction time and risk behavior when infected. But these findings are narrow, tentative, and often limited to specific conditions under laboratory testing. They do not translate into broad superhero-style immunity in everyday life. Rh-negative still catch colds. They still get viruses. They still end up in hospital wards and waiting rooms like everyone else. The gap between subtle statistical differences in a lab and I am immune to entire categories of disease is wide. Much of what gets repeated online belongs on the second side of that gap. From here, the claims start to leave biology entirely. One of the strangest myths is the reptilian body temperature idea. In the story, Rh-negative individuals are said to run colder like reptiles or to be less able to regulate their temperature. Some tie this to ancient alien or non-human ancestry, blending conspiracy theories with pop culture references until the original idea is almost unrecognizable. In real physiology, human body temperature varies. Some people naturally run a bit cooler or warmer. Stress, hormones, time of day, and infection all shift the needle. A few studies have found that Rh-negative individuals may on average have slightly different thermal comfort zones, but nothing that turns them into a separate class of organism. The reptilian label doesn't come from a lab. It comes from an older symbolic language in which reptiles are mysterious, cold-blooded, alien. Calling Rh-negative bodies reptilian is less about temperature and more about storytelling. It paints them as otherworldly, different, not just in degree but in kind. And once the door to the otherworldly is open, more claims enter. Psychic sensitivity, telepathy, stronger empathic perception, a sense of knowing things before they happen, an uncanny knack for sensing danger or reading people. If you listen quietly, you'll hear recurring patterns in these stories. I always knew when the phone was going to ring. I dream things and then they happen. I walk into a room and feel overwhelmed by everyone else's emotions. I just know when someone is lying. On their own, these experiences are not unique to Rh-negatives. People of every blood type and every culture have stories of intuition, déjà vu, prophetic dreams. Our brains are pattern-detecting machines. They log thousands of impressions every day, many beneath conscious awareness. When a few of those impressions line up with later events, the moment feels magical. But if before that moment, you've already been told you're different, that your blood is rare, that your body runs on unusual settings, then every intuitive hit becomes evidence, proof that the stories might be true. The identity circularly confirms itself. So, what does science actually say about Rh negatives and psychic ability? So far, nothing solid. No controlled, replicable studies have shown enhanced extrasensory perception tied to the Rh factor. What has been more consistently observed is that people who already believe in paranormal phenomena are more likely to interpret ambiguous experiences as supernatural. They're also more likely to seek confirming information, to join communities of like-minded people, and to notice patterns that fit their expectations. This doesn't mean their experiences are fake. It simply means we do not have evidence that the Rh antigen sitting quietly on the surface of red blood cells is the master switch controlling them. So, why then are these ideas so persistent, so compelling? Part of the answer lies not in biology, but in psychology. Being Rh negative can feel like belonging to a small hidden club. Only about 15% of people worldwide share this trait, and in some regions the number is far lower. Rarity invites meaning. When something about you is uncommon, it's natural to ask why. In a complex world where identities shift and traditional markers of belonging, village, religion, extended family are often weakened, a biological label can feel solid, tangible, anchor-like. This is in my blood. This is what I am. Blood-type identity theories, whether in Japan's personality system or in Rh-negative superpower stories, offer simple narratives in a confusing universe. They tell you who you are, why you feel different, and where you belong. I'm not just shy, I'm this type. I'm not just sensitive, my bloodline is special. I'm not just anxious or observant, I'm an empath, a seer, a leftover from some older, purer group. These stories can be comforting. They can bring people together who might otherwise feel alone. They can spark curiosity about genetics, history, and ancestry. They can even serve as metaphors, ways of talking about temperament and experience in a language that feels vivid and alive. The risk comes when metaphor hardens into dogma. When someone is told that their blood type limits who they can love, what job they can do, or whether their child will be normal, when health advice is based more on rumor than evidence. When a rare trait is used to elevate one group of humans above another, the challenge then is balance. To recognize that some correlations are real but small, that many claims are untested, and some are almost certainly incorrect. That feeling different does not require a supernatural explanation, even if it sometimes longs for one. And yet, it is also true that science is not finished with our blood. Large-scale genetic studies are continually uncovering subtle links between blood factors and disease, behavior, and physiology. As tools improve, researchers may yet find unexpected patterns involving Rh status, effects too delicate for earlier generations to detect. Keeping an open mind does not mean believing every story. It means being willing to revise your understanding when solid evidence appears, and equally willing to set aside ideas that don't survive careful testing. So, when someone says, I'm Rh negative, and I feel like I'm more intuitive, more sensitive, more something, there are two ways to listen. One way is to take their list of abilities as literal blood-coded superpowers. The other is to hear it as a description of personality, of lived experience, of a lifelong search for an explanation of why they feel the way they do. In the quiet space of late night, with the lights dim and the world fading to a softer focus, it can be strangely soothing to imagine that a part of you carries ancient secret qualities, that your blood holds echoes of forgotten lineages, strange climates, lost migrations. Maybe some of that is true in ways we don't yet fully understand. Maybe much of it is poetic rather than literal. Either way, the red cells circulating silently through your veins tonight are doing something undeniably extraordinary. They're keeping you alive, feeding your brain, fueling your heart, linking you through a chain of unbroken descent to ancestors who survived plagues, famines, ice ages, and childbirth in eras when survival was anything but guaranteed. Whether your Rh factor is positive or negative, that alone is a kind of superpower. One shared remarkably by every human being still breathing on this planet. And the story of what your blood might say about your personality, your destiny, your health continues into the next chapter. Picture a quiet office in Tokyo. Fluorescent lights humming softly, the click of keyboards, the low murmur of voices. It's a perfectly ordinary workplace until the manager during a team-building session casually asks everyone to write their blood type on a name tag. Nobody flinches. Nobody thinks it's strange. For many people in Japan, that tiny detail, A, B, O, A, B, isn't just a medical note. It's a personality profile, a hint about whether you're steady or impulsive, shy or outgoing, a good team player or a loose cannon. This is blood type personality theory. According to this system, people with type A blood are supposed to be earnest, organized, considerate. Type B are said to be creative, individualistic, sometimes selfish. Type O are painted as confident, natural leaders. Type AB are mysterious, complex, a mix of extremes. It's a full-blown typology, like a zodiac made of red cells and plasma instead of stars and planets. Walk into a bookstore in Japan and you can find blood type compatibility guides, blood type diet books, blood type parenting manuals. Television shows joke about celebrity blood types. Pop idols might mention theirs in interviews the way someone in the West might casually mention their zodiac sign. And here's the fascinating part. Underneath all of that, they're talking almost entirely about the ABO system, not the Rh factor. The focus is on A, B, O, and AB, the surface markers on red blood cells first classified in the early 1900s. The plus or minus that tells you whether you're Rh positive or Rh negative rarely enters the story. To someone fascinated with Rh negative blood in the West, this can feel almost upside down. In many English-language conspiracy forums or fringe science books, the ABO groups are just background. The real mystery, the alleged alien gene or ancient bloodline, is often pinned on Rh negative status, not A, B, O, or AB, but the minus sign. Two cultures staring at the same blood and choosing completely different places to hang their myths. In Japan, the blood type personality system has roots in early 20th century pseudoscience. A 1920s-era researcher tried to link blood type with racial character, echoing dangerous ideas that were circulating in Europe at that time. Those early theories were discredited after the war, but the notion lingered in pop culture, slowly mutating from politics into entertainment, from ideology into icebreaker. By the 1970s and 80s, blood type personality books became bestsellers. Cartoons, magazines, talk shows, they all played with the idea that your blood type could explain your quirks, your romantic compatibility, your lucky days. It was lighthearted, catchy, easy to remember, something you could bring up on a first date or in a schoolyard conversation. But ideas, even playful ones, have consequences. In Japanese, there's a word birahara, short for blood harassment. It describes discrimination based on blood type. The colleague who doesn't get put in charge of a project because type Bs can't be trusted with details. The child who's teased at school for being too serious, just like an A. The manager who prefers to hire type O for sales because they're natural extroverts. These judgments don't show up on ancient tablets or in religious texts. They come from mid-20th century magazines, self-help books, and pop psychology. But for the people who experience them, they can be very real. In some workplaces, applicants have reported being asked their blood type during hiring. In some dating services, profiles quietly include it. Not everywhere, not all the time, but often enough that government agencies and scientists in Japan have issued public statements reminding people that there is no scientific basis for blood type personality theory. That an A on a form doesn't tell you whether someone is kind or cruel, loyal or deceptive. It only tells how their immune system will react to a transfusion. Meanwhile, on the other side of the world, a different story is being told. In many Western countries, especially in online niches, interest in blood and personality takes a more specific turn. It doesn't stop at ABO types. It dives into Rh negative. Rh negative is sometimes portrayed not as a simple variant, but as a hint of something extraordinary. People claim that Rh negative individuals are more intuitive, more empathetic, more independent, more prone to vivid dreams, more resistant to certain diseases, more likely to feel like outsiders, like they don't quite fit in. Scroll through forums and you'll find long lists of common Rh-negative traits. High IQ, piercing blue or green eyes, sensitivity to light, psychic abilities, strong sense of justice. The lists are rarely consistent, but they share a common thread. The idea that Rh-negative blood marks you as different, special, set apart from the crowd. This is not the Japan-style personality typology with neat categories for A, B, O, and AB. It's something closer to a myth of chosenness. Not everyone has Rh-negative blood. In some populations, it's very rare. That rarity becomes a canvas. If you're one of the few, you might feel like you've discovered a secret identity, a hidden badge you've carried your whole life without knowing it. Of course, scientists have studied blood groups and personality, not because they hope to prove magic, but because they wanted to be thorough. If blood type affects disease risk, maybe just maybe it could be linked to certain behavioral tendencies through subtle pathways, metabolism, hormones, brain chemistry. Hundreds of studies have looked for patterns. Are type A's more anxious? Are type O's more outgoing? Do Rh-negative score differently on personality inventories? The results are remarkably consistent in one way. There's no robust reproducible evidence that blood type determines personality in any meaningful sense. Individual studies sometimes find minor correlations, a tiny bump here, a slight tendency there. But when larger analyses combine the data, the signals fade into noise. If blood type has any effect on personality, it's so small that it's drowned out by everything else: upbringing, culture, life experience, individual genetics, chance. Professional organizations in psychology and medicine, particularly in Japan, have publicly stated that blood type personality theory isn't supported by strong science. It lives in the realm of culture, not biology. A story we tell about ourselves rather than a fact written in ourselves. And yet the story persists both in the East with personality typology and in the West with Rh-negative mystique. Why? Because humans are drawn to categories. For hundreds of thousands of years, your ancestors survived by sorting the world into patterns. Safe and dangerous, friend and stranger, edible and poisonous. Over time that instinct expanded. We started sorting ourselves. Tribe and outsider, family and rival, clan and clan. In a small band of hunter-gatherers, you might know everyone personally. You'd remember who was generous, who was moody, who told the best stories by the fire. But as societies grew, we needed shortcuts, ways to make quick guesses about strangers. Sometimes those shortcuts were visual clothing, tattoos, hairstyles. Sometimes they were abstract astrology signs, clan names, totems. In the modern world, they can be anything from your favorite sports team to your Myers-Briggs type, to your Hogwarts house. Blood type fits perfectly into that ancient instinct. It's invisible, mysterious, and yet neatly labeled. One letter, one sign, a tiny fragment of biology that can be inflated into a whole personality sketch. It gives us something we crave: identity. If you're a type A in Japan, you can read about your traits and feel seen, even if the description is generic enough to apply to almost anyone. If you're Rh-negative in Europe or America, you can stumble across an article about the rare blood of the chosen ones and feel a rush of recognition, even if the details are vague. Psychologists have a name for this tendency: the Barnum effect. The habit of seeing ourselves in broad, flattering descriptions and thinking they're uniquely tailored to us. You're sometimes shy, but you can also be outgoing in the right situation. You care about others, but you wish they would understand you better. You're creative, but people often misunderstand your depth. Vague enough to fit almost anyone, specific enough to feel personal. Blood type personality theories plug directly into that vulnerable, hopeful part of us that wants to be understood without having to explain ourselves. They offer an instant story. This is who you are. This is why you feel the way you do. This is why you struggle, why you succeed, why you don't quite fit the mold. And for Rh-negatives, there's an extra twist: rarity. Most people in the world are Rh positive. Rh-negative is the minority, in some regions a tiny one. Being part of a small group can be frightening. You're different, you're unusual, but it can also be comforting if someone offers you a narrative that makes your difference meaningful. Instead of you're rare and that's random, the story becomes you're rare and that means you're special. Maybe descended from ancient rulers, maybe shaped by unusual evolutionary pressures, maybe carriers of traits that the world will one day need. From a scientific standpoint, Rh-negative is simply one variant among many, a quirk of genetics tied to a protein on red blood cells. It has real medical implications, especially in pregnancy, but no confirmed link to intelligence, ESP, empathy, or spirituality. Yet, on a psychological level, it can function like a quiet, secret club, a way to say, I belong somewhere. There are others like me. It's easy to dismiss these beliefs as superstition, but look a little closer and they reveal something very human. Underneath the pseudoscience and the sensational headlines lies a universal longing to find patterns in chaos, to locate ourselves in a story grander than one life. To feel that our blood doesn't just keep us alive, but connects us to something. In Japan, that something might be a shared cultural game, a playful way of bonding with coworkers or classmates. In Western Rh-negative circles, it might be a sense of being part of an ancient thread, a lineage that survived hidden and obscure. In both cases, science quietly stands to the side, reminding us the molecules in your blood don't dictate your kindness or cruelty, your courage or your fear. They don't predetermine your destiny, but the stories you tell about the blood, those can shape how you see yourself, how you move through the world, what you feel entitled to, afraid of, proud of. And so, when you hear someone say, "Oh, that's so type A of you," or, "Of course you're Rh-negative," that explains everything. You're not just listening to a casual remark, you're hearing echoes of a much older habit. The ancient urge to take the messy, unpredictable complexity of human beings and file it into neat little boxes. Useful sometimes, harmful sometimes, comforting often because in the end, whether your blood is A, B, O, or AB, positive or negative, it carries the same fundamental story, a chain of ancestors who adapted, and passed life forward, drop by fragile drop. And that story, the one written in survival rather than stereotypes, is far more astonishing than any personality chart. Still, myths have power, and the myth of blood and character is not done yet. As medicine learns more about how blood types influence disease risk, and as culture continues to spin new narratives around old molecules, the line between biology and identity will be tested again and again. The question is not just what your blood can do, it's what you choose to believe it means. Fast forward from Ice Age river valleys and vanished human cousins to something utterly modern. Fluorescent hospital lights, plastic tubing, the quiet, urgent ballet of an emergency room. At 3 a.m. a paramedic bursts through the door with a patient on a stretcher. Car crash. Massive blood loss. The clock is ticking in minutes, maybe seconds, before anyone knows this person's name, before their medical history, before their story. The staff needs to know one thing. What kind of blood can we safely put into their veins? This is where Rh-negative blood quietly steps out of the shadows of genetic mystery and becomes something else. A medical lifeline. In a crisis, you can't wait for the lab to run a full cross-match. You need a blood type that will almost never trigger a deadly reaction. For decades, that break glass in case of emergency blood has been O-negative. In many hospitals, if you walk into the blood bank, you'll see one shelf treated almost like gold, labeled in bold letters, carefully inventoried. That's the O-negative stock, the donors the staff know by name, the units they hesitate to use, unless they absolutely have to. Because in the high-speed triage logic of modern medicine, O-negative is as close as we get to a universal key. But the story is more complicated than the slogan you might have heard in a high school biology class. The universal donor myth makes it sound simple. O-negative goes into anyone, everyone, no problem. The reality is filled with small print. Blood compatibility isn't just about A, B, AB, and O. It isn't even just about positive and negative. Human red blood cells carry dozens of antigen systems. Kel, Duffy, Kid, MNS, and others with names that sound like obscure characters from a forgotten novel. Each system can in theory trigger an immune response. What made O-negative famous is that on the two biggest and most dangerous systems, the ABO group and the RHD factor, it's stripped down to the bare minimum. No A antigens, no B antigens, no RHD antigens. To most immune systems, O-negative looks like the least threatening stranger in the room. So in emergencies, when there isn't time to figure out exactly who you are in the language of blood, doctors reach for the least suspicious option. But even O-negative isn't truly universal. Some patients carry rare antibodies against those smaller, more obscure antigens. For them, even O-negative can be dangerous. That's why outside of immediate life or death situations, hospitals still insist on careful matching. Test, confirm, double-check. The quiet, methodical side of medicine, humming in the background while the drama plays out in the operating room. Still, when seconds matter, O-negative saves lives over and over again, which is why the people who carry that blood type, especially O-negative, quietly shoulder a special kind of medical responsibility. In a way, the rarity that makes their blood historically mysterious also makes it logistically difficult because there's a problem. There's never enough of it. Imagine a city where 15% of the population speaks a rare language. Now imagine that in times of crisis, natural disasters, accidents, sudden illness, that language is the only one certain people can understand. You'd want plenty of translators on hand, but in the real world of blood banks, those translators are almost always in short supply. Rh-negative blood types, including O-negative, are globally rare. In some populations, fewer than 5% of people are Rh-negative, yet those are the units hospitals need to stock, not just for emergencies, but for every Rh-negative patient who comes through the door. If you're Rh-negative, you can't safely receive Rh-positive blood without risking sensitization. Your immune system recognizing the Rhd antigen as foreign and building a memory response. That's especially dangerous for women of childbearing age because of the pregnancy complications we explored earlier. So, while most people can accept either positive or negative of their own ABO type in a pinch, Rh-negative patients are stuck in a narrow lane. For them, the supply line is brutally simple. No Rh-negative donors, no compatible blood. This creates a quiet, constant tension in hospital blood banks. They track their Rh-negative inventory like a slow-burning fuse. They know how fast it's used, how long it takes to replace, and how catastrophic it would be to run dry at the wrong moment. Blood donation organizations have learned to live with this. You'll sometimes see targeted campaigns. O-negative donors urgently needed, phone calls, texts, that subtle sense of being permanently on a list. To be Rh-negative in the modern world is to carry a hidden logistical value. Your blood is not just another data point. It's a scarce resource rationed, planned, and sometimes begged for. But the Rh-negative story in medicine isn't only about supply and demand. It's also about something even stranger. Possible differences in disease risk. For most of the 20th century, doctors knew blood groups mattered in transfusions and almost nowhere else. Then researchers started noticing patterns. People with blood type O, for example, tend to have a slightly lower risk of certain blood clot-related events, but a slightly higher risk of severe bleeding after trauma. Type A has been linked in some studies to a higher risk of certain cancers or heart disease. None of this is simple and none of it is destiny. But the patterns kept appearing. The Rh factor got less attention at first. It was mostly a pregnancy problem, a transfusion issue. But slowly studies began hinting that Rh-negative status might nudge the odds in subtle ways. There have been suggestions, for now still tentative, that Rh-negative individuals might have slightly different risks for some infections, allergies, or autoimmune conditions. Some workers looked at Rh factor and susceptibility to parasites like Toxoplasma gondii. Other studies have examined links between Rh type and mental health traits, reaction times, or even resistance to certain kinds of severe malaria, though the evidence is still patchy. Then came 2020. A new virus, SARS-CoV-2, circled the globe. Hospitals filled again, not with trauma victims this time, but with patients struggling to breathe. Somewhere in the chaos, scientists turned once more to an old question. Does blood type matter? Early studies, especially from China and Europe, hinted at something intriguing. People with blood type A seemed, in some data sets, to have a slightly higher risk of severe COVID-19. People with type O seemed to have a slightly lower one. As for Rh factor, several analyses suggested that being Rh negative might be associated with a modest reduction in risk of infection or severe disease. Not immunity, not a force field, just a gentle tilt in the odds. As more data accumulated, the picture became more complicated. Some studies supported the association. Others found weaker effects or none at all. Once age, underlying health, and exposure patterns were accounted for, the virus was new, the statistics were messy. Human lives rarely line up neatly with the categories we invent. By late 2021, the consensus was cautious. Yes, there might be small correlations between blood type, including Rh status and COVID-19 risk, but they were minor compared to bigger factors. Age, obesity, chronic illness, access to health care, vaccination. Still, the idea lingered in the public imagination. People searched their blood donor cards, typed questions into search engines late at night. Am I safer? Am I at more risk? The more scientifically grounded answer is both reassuring and humbling. If there is an Rh negative advantage against COVID-19, it's likely small, a nudge, not a shield. Interesting to researchers, but not decisive for individuals. And yet, from the perspective of evolution and epidemiology, even small differences can matter. A fraction of a percent multiplied over millions of people and multiple generations can shift the arc of history just a tiny bit. It's possible that in past pandemics, long before modern records, certain blood types, Rh negative among them, fared slightly better or worse. We may never know. Those stories are buried with our ancestors, written only in survivorship curves that have long since flattened into the present. While scientists sift through statistical noise, looking for faint signals of advantage, another frontier is emerging that could one day transform the whole conversation. Synthetic blood. For now, every unit of transfusible blood comes from one place, another human being, screened, tested, typed, and stored. It's a fragile, perishable system. Blood has a shelf life. It must be kept cold. It can't be stockpiled forever. This creates vulnerabilities, natural disasters, wars, pandemics, seasonal shortages, and for rare blood types, including some Rh negative variants with unusual antigen patterns, the problem is even sharper. Occasionally, a patient needs a match so specific that only a handful of donors in an entire country are compatible. So, researchers have been working on alternatives. Some efforts focus on blood substitutes, solutions that can carry oxygen like hemoglobin-based products or perfluorocarbon emulsions. Others look at the problem from a different angle, growing red blood cells in the lab. Imagine taking stem cells, primitive flexible cells that can become many different tissues, and coaxing them to mature into red blood cells in bioreactors. Identical, standardized, batch-produced. If you engineer them to lack certain antigens, you might create a truly universal red cell as close to invisible to the immune system as possible. There have already been small human trials of lab-grown red blood cells. Not science fiction, but early careful steps. The challenges are enormous. Cost, scale, safety, regulation. Turning a delicate lab process into something that can fill hospital refrigerators around the world is no small feat if it works. If one day synthetic or lab-grown blood becomes routine, what does that mean for O-negative individuals? In one sense, it could relieve the pressure. No more chronic shortages. No more panic when the O-negative shelf starts to empty. Rare blood would no longer hold the same logistical power. But in another sense, the Rh-negative story wouldn't end at all. Because to build synthetic blood systems that work for everyone, scientists need genetic and immunological reference points from real human donors, including the rare ones. Rh-negative blood with its stripped-down antigen profile is a crucial template. It helps researchers understand how to minimize immune reactions, how to design cell surfaces that read as safe. And if we ever want to biomanufacture extremely rare antigen combinations for patients with complex transfusion histories, those recipes will have to be written from the blood of people who carry those traits today, which brings us to the quiet, practical part of the Rh-negative advantage, the part that doesn't involve ancient aliens, royal conspiracies, or hidden superpowers. The part that simply asks, what can you do right now with the blood you carry? If you're O-negative, especially O-negative, You are part of a small, globally scattered minority whose biology is disproportionately useful to medicine. Every donation you make has a high statistical chance of ending up in an emergency situation, a neonatal intensive care unit, an operating room where doctors are running low on options. If you choose to register as a regular donor, you become part of a living infrastructure, a quiet, ongoing act of community survival, a continuation in a strange, modern form of those ancient networks of cooperation that carried our ancestors through climatic catastrophes and population bottlenecks. Beyond donation, Rh-negative individuals can volunteer for medical research studies, genetic sequencing projects, immunology trials, population health surveys. When scientists design studies about blood types and disease risk, they need enough Rh-negative participants to draw meaningful conclusions. Without them, the data is forever lopsided. Sometimes contributing is as simple as answering a questionnaire. Other times, it might mean a vial of blood for a lab to analyze, a few minutes in a clinic, a small inconvenience that feeds into a much larger story. Because in the end, the Rh-negative advantage in modern medicine isn't a superpower you feel in your bones. It's not faster healing or X-ray vision. It's something subtler. It's the way your blood fits into the puzzle of human survival at a moment in history when we finally understand in molecular detail just how fragile that puzzle really is. It's the role your rare antigen pattern plays in emergency transfusions, in blood bank logistics, in cutting-edge labs where scientists are trying to grow the future in glass flasks and steel tanks. It's the fact that simply by existing, you carry forward a genetic variation that has threaded its way through millennia of births and deaths, plagues and famines, migrations and wars to arrive in a world of white coats and centrifuges and softly beeping monitors. A world where the same invisible markers that once might have shaped life and death on a prehistoric savanna now shape life and death under LED lights and sterile drapes. The Rh-negative mystery began long before hospitals, but in modern medicine, it has found a new stage, not as a cosmic anomaly, but as a working tool, a rare resource, a bridge between ancient genetics and present-day survival. And even here in the clinical brightness of science, the questions haven't stopped. If anything, they've multiplied. Because the more we use this rare blood, the more we study it, the more we ask it to teach us about disease, immunity, and human variation, the more we realize how much we still don't know. So, where does all this leave us? After a century of blood typing, decades of genetics, thousands of papers and millions of donated samples, what do we actually know about Rh-negative blood? And what still sits out there in the dark, just beyond the edge of the campfire? Let's start with the part that isn't a mystery at all. Rh-negative blood is not alien. It is not proof of secret royal hybrids, underground civilizations, or visitors from the Pleiades. Genetically, we can see exactly what it is, a missing protein on the surface of red blood cells, a deletion, a disruption, a genetic edit in the RHD gene. But that blunt technical description hides a deeper question. Yes, we know what it is. We still don't fully know why it spread the way it did, or why it's clustered so strangely in certain populations. That's the first real mystery. Why do some isolated groups in Europe show Rh-negative frequencies above 30 or even 40% while vast regions of Asia and Africa barely register a few percent? Why does this one little missing protein form such a sharp genetic border between neighboring populations that have traded, migrated, and intermarried for thousands of years? We have good theories. Maybe it's random genetic drift magnified in small ancient communities. Maybe some past disease long gone and forgotten once made Rh-negative carriers slightly more likely to survive. Maybe it's tied to environmental factors we haven't even recognized yet. But the key word is maybe. We don't yet have the kind of clean, decisive evidence scientists love. No fossilized microbe we can point to and say, ah, this pathogen, this is why. No ancient DNA time series that shows Rh-negative suddenly shooting up after a particular epidemic. Just hints, patterns, probabilities. That's the second mystery. We know roughly when the main Rh-negative variants appeared in the human lineage. Genetic clocks, comparisons of ancient remains, and modern populations all converge on the idea that these mutations are relatively recent on an evolutionary scale, probably tens of thousands of years old, not hundreds of thousands, but roughly is doing a lot of work. We're still missing big parts of the timeline. We don't yet have dense ancient DNA coverage from every corner of the world, every small valley and mountain pass where strange things might have happened to our ancestors. We have snapshots, not a movie. And that's where the limits of our tools begin to show. When you send your saliva to a commercial ancestry test, the company looks at scattered markers, tiny signposts across your genome. They're not sequencing every base, not reconstructing every mutation you carry. They're doing a kind of genetic sampling, good enough to guess your ancestry in broad strokes, but not detailed enough to capture the full story of a trait like Rh status. Even in research labs with full genome sequencing and powerful algorithms, we're still staring through fog. We can see that certain Rh-negative lineages cluster in some regions. We can infer that some versions likely arose independently more than once. We can trace how they spread and mixed and faded as populations moved, but we can't yet scroll backwards through time and watch the very first Rh-negative child being born somewhere in prehistory to parents who had no idea that their baby's blood carried a tiny difference that would echo into modern conspiracy theories. That's the third mystery. We still don't know the full set of consequences that come along with being Rh-negative. For years, people have collected anecdotes. I'm Rh-negative and I never seem to get this or that disease. I'm Rh-negative and my body reacts strangely to anesthesia. I'm Rh-negative and I feel like I'm always cold. Pieces of a puzzle or just noise. So far, large-scale studies suggest that aside from pregnancy issues and transfusion compatibility, Rh status doesn't radically alter health outcomes for most people, at least not in ways that are huge and obvious. But science thrives on large numbers, and Rh-negative is rare. In many regions, there simply aren't enough Rh-negative individuals in existing data sets to detect more subtle effects, a slight tilt in immune response here, a small shift in susceptibility there. A difference that only shows up under specific environmental conditions. Those things are hard to catch, so we're left with a mix of solid facts and unexplored shadows. And that's exactly the space where human imagination wakes up, because Rh-negative blood sits at a strange intersection. It is visible, testable, mapped onto real people's bodies. It shows up in hospital charts, ancestry graphs, and genetic diagrams. It affects real pregnancies, real transfusions, real emergencies. But it's also rare, unevenly distributed, historically entangled with myths of pure bloodlines, royal families, ancient tribes, and secret societies. It feels special. If you're Rh-negative, you might have discovered it not in a textbook, but in a moment of quiet shock sitting in a doctor's office hearing that your blood could attack your own unborn child without medical help, or hearing that your blood type is universal donor for emergencies needed when time is running out. Those experiences carve deep emotional grooves. Later, you might stumble across strange stories online, claims that Rh-negative people are more intuitive, more sensitive to temperature, more prone to unusual dreams, claims of higher IQs or lower. Some say they get more nosebleeds. Others say they feel different in crowds. A few describe feeling out of place, as if they don't quite belong to their family, their culture, even their own era. Are those patterns real or just the human tendency to weave meaning out of coincidence? Science, when it is being honest, has to say we don't know for most of these claims. To test them properly, you would need huge, carefully controlled studies with thousands of Rh-negative and Rh-positive people matched for age, culture, lifestyle, and so on. You'd need to separate psychological suggestion from biology, myth from measurement. For now, we mostly have stories, quiet, late-night stories. The woman who swears that every time she donates blood, something in her body feels deeply, oddly right, as if she is doing what her ancestors did in some forgotten crisis. The man who grew up in a region where almost everyone is Rh-positive, who was told as a child that his blood was strange and special, and who still feels a faint distance from the people around him. The mother who lost a pregnancy before Rh disease was fully controlled in her country, and who still thinks of her blood as both miraculous and dangerous, life-giving and life-threatening at the same time. Each story is one data point in a vast, uncharted landscape of human experience. And that brings us to the balance we have to strike. On one side, there is mystery, the real, valuable kind, the recognition that we don't yet understand everything. That human biology still has hidden corners and unasked questions. On the other side, there is evidence, the commitment not to leap from we don't know to anything is possible. Therefore, everything is true. Being intellectually honest means holding both at once. It means we can say, yes, it is genuinely strange that a blood factor that can make pregnancy so dangerous wasn't immediately wiped out by natural selection. That suggests there might be some hidden advantage now lost or subtle that kept it in the gene pool. Yes, it is genuinely interesting that Rh-negative frequencies form sharp geographic patterns, especially in regions like the Basque country, where genetic histories are already unusual. Yes, it is genuinely worth studying whether Rh status correlates with certain immune responses, disease risks, or even neurological traits. And at the same time, we can say no, we do not have solid evidence that Rh-negative people are more likely to have psychic abilities, secret powers, or cosmic destinies. No, we do not have proof that any specific ancient civilization was Rh-negative only or that ruling classes systematically bred for Rh-negative status to maintain some occult advantage. No, we cannot turn a single missing protein into a magical explanation for everything that makes a person unique. Because in the end, Rh-negative is just one note in a vast genetic symphony. Your blood type is a tiny fraction of who you are. You carry millions of genetic variants, traces of bottlenecks, migrations, plagues, famines, and forgotten loves stretching back through the ages. Your eyes, your brain, your immune system, your bones, your temperament, all are shaped by layers of ancestry and experience that go far beyond a plus or minus sign on a lab report. And that might be the real lesson hidden inside this whole mystery. Rh-negative blood reminds us how wonderfully irreducibly complex human diversity really is. One small molecular detail absent on the surface of a red blood cell turns into a story that crosses medicine, anthropology, history, and myth. It shapes who can safely receive your blood, how your pregnancies must be monitored, what ancient population you might be distantly connected to. But it also shows us our limits. Even with all our modern tools, we still can't answer every why. We can trace the pathways, map the frequencies, model the probabilities. Yet, the deeper motives of evolution often remain half-hidden, like footprints fading into mist. Still, the story isn't over. In the coming decades, as more people around the world have their genomes sequenced, the statistical power to study rare traits like Rh-negativity will grow. We may finally be able to detect subtle health effects, slight differences in infection resistance, or in responses to certain medications. New techniques in ancient DNA extraction may push further back into time, pulling fragments of genetic information out of bones that have lain in the earth for tens of thousands of years. We might someday watch Rh-negative variants appear and spread across prehistoric landscapes like embers carried by the wind. We may discover that Rh-negative was favored in some epidemic we've never identified or in some ecological niche now vanished. We may find that for a few thousand years in one particular region, Rh-negative was a quiet survival advantage, a tiny edge that meant more grandchildren, more great-grandchildren, and eventually more of that gene in the population, or we may find something even stranger, that it never had a big advantage at all, that it survived simply because evolution is messy. Because chance events in small populations can lift some traits and bury others for no deep reason at all. Sometimes the story written in our blood is one of brutal necessity. Sometimes it's one of exquisite accident. For now, Rh-negative blood sits between those possibilities. A real biological anomaly, thoroughly mapped and yet not fully explained. A trait that can complicate pregnancy and yet persists. A marker that ties together remote mountain villages, coastal towns, and modern cities in quiet invisible kinship. And maybe that's the thought to fall asleep with. Not that Rh-negative blood makes you more than human or less, but that it is one more thread in the vast intricate tapestry of what it means to be human at all. One more reminder that inside every one of us beneath the skin and the stories, the old secrets of our species are still flowing. Beat by beat, cell by cell, waiting for future generations to read them more clearly. Until then, the mystery endures, half solved, half hidden, circulating in silence through millions of hearts around the world. So now here you are lying in the dark, breathing slowly, listening to a story that began long before there were nations or cities or even names. A story written in something you carry with you every second of your life, your blood. We've wandered from ancient bottlenecks and vanished cousins to strange antigens and silent pregnancies. From queens with blue blood to lab technicians staring in confusion at test tubes that refuse to behave. From legends of alien origins to gene variants that quietly help fight disease, all of it flowing through one simple question. What does it mean to have a blood type that almost shouldn't exist? And yet the real twist is this. Rh-negative might feel rare, mysterious, special, but underneath all those labels, it's still part of the same crimson river that runs through everyone. The same fused chromosomes born in a long-ago crisis. The same ancient survival toolkit refined by 100,000 years of near extinction and slow recovery. Whether your blood is Rh positive or Rh negative, it's all a different pattern on the same tapestry. Your heart doesn't care about internet theories. It just keeps pumping. Every beat is an echo of those first small bands huddled around fires, sharing stories under unfamiliar stars. Every drop carries memories of Neanderthals hunting in the snow, Denisovans crossing dark oceans, early Homo sapiens tracing rivers out of Africa. All of that condensed into the quiet pulse in your wrist. Some mysteries are out there in the universe written in distant galaxies and collapsing stars, but some of the deepest ones are much closer inside your veins. In the way one tiny protein on the surface of a red blood cell can decide whether a pregnancy is easy or deadly. In the way a rare Rh-negative mother can carry a child whose blood her own body wants to reject. And how modern medicine can gently persuade the body to choose mercy instead. If nothing else, Rh-negative blood is a reminder that you are not simple. You are not ordinary. Even if your blood type is the most common in the world, the fact that it works at all is astonishing. Millions of red cells flowing in perfect rhythm. Hemoglobin catching oxygen in your lungs, letting it go in your fingertips. Ancient genetic gambles paying off in the quiet luxury of you simply being alive tonight. When this video ends, you might forget the name of a particular antigen or how the Rh factor was first discovered, but maybe you'll remember this. Your body is full of unsolved puzzles. Small everyday miracles that scientists are still trying to understand. You are quite literally a walking archive of evolutionary experiments and family stories reaching back hundreds of thousands of years. If you feel like taking the mystery a step further, you could look up your own blood type if you don't already know it. Ask your parents or grandparents what theirs is, trace the patterns. See how something as invisible as a protein on a cell connects relatives who lived and died long before you were born. A quiet family map drawn in red. And if tonight's journey kept your mind gently wandering while you drift towards sleep, you can support this little corner of curiosity in a few simple ways. You can tap the like button, not as a big dramatic gesture, just as a soft signal that you'd like more stories like this. You can subscribe if you'd like to keep exploring hidden chapters of history, science, and myth while you rest. I'd love to know where in the world you're listening from. If you feel awake enough, leave a comment with your country, your city, or just listening from bed and your time zone. It's a quiet kind of magic, seeing how many different places are connected by the same story at the same moment through the same glow of a screen in the dark. For those who want to go a little further in supporting the channel, there's an optional membership here on YouTube, completely up to you. No pressure at all. Think of it as a way to help keep these long, gentle, deep dives coming. So, there's always another story waiting when you need help falling asleep. For now, you don't have to do anything. You don't have to remember every detail or pick a theory or solve the mystery of Rh-negative blood. You can just let it all settle. Let the images fade. Ancient fires, frozen valleys, silent laboratories, hospital rooms where a tiny vial of medicine turns a deadly incompatibility into a healthy birth. Feel your own heartbeat, steady, patient, older than any legend. The blood that puzzled scientists, the blood that fueled empires, the blood that defied extinction. It's all the same story, all the same river. And tonight, it carries you gently towards sleep. Rest well. May your dreams be curious, your thoughts be soft, and your body remember in its own quiet language that you are part of a lineage of survivors stretching back almost 1,000,000 years. Good night.